Good morning and good afternoon. Thank you for joining our webinar series today. This is the start of our nCounter Masterclass Series. My name is Tim Wesselman. I'm the CEO, President, and founder of Rosalind. It's my pleasure to be here today and to introduce our key speaker, who's our Vice President of Customer Experience at Rosalind. I'm going to talk more about Ryan's background here, and I want to make a few other comments as we transition. Not only does Ryan lead our customer experience team, which includes customer success scientists, but also our customer support team. Ryan holds a PhD in bioengineering from UCSD. He has spent years at NanoString. He has been in your shoes. He has been a wet scientist in the lab. He has analyzed nCounter data as well as many other types of data. He understands entirely what you go through. This is going to be an incredible series. We typically reserve this for our enterprise and professional users. We've opened this up for everyone. Really looking forward to having Ryan go through all of this. I'm going to make just a few quick housekeeping comments. As a reminder, this is GoToWebinar. You're all muted, so don't worry about your audio. Please hold your questions if you want to ask them till the end, but you can type them in. So when they come to you, just type them in the little question box on the side, and I'll be working with Kuki, who's our Director of Product and Business Strategy. She'll be working with me to gather up the questions, and we'll all answer those for you at the end. We did add a couple really nice handouts that are attached. Please look for those and download them. And I'll just make one other quick comment here. First to mention, our whole objective and where I try to drive this team is to help you all achieve the outcomes that you're looking for in your research, whether that's a publication, whether it's identifying a new target. We're focused on helping you find answers in your data. And one quick bit about NanoString. We really love this partnership with NanoString, and we love the nCounter instrument. And a super quick story, I had a chance to sit with an executive from J&J just in the last few days. And we were talking about how in our industry, sometimes we lose sight of the fact that less is more. And with a focus panel from nCounter, US scientists are able to get highly sensitive, highly accurate results without the distraction of other background that could create noise, which is a big focus for today, and really get sensitive, immediate results between the instrument and the Rosalind platform. And Ryan's going to explain all of that to you today. So kicking off the Masterclass series, Dr. Ryan Friese, over to you.
Thank you, Tim. I appreciate that introduction. I'm very excited to lead this masterclass. This idea has been floating around my head for years. I want to share all the knowledge I've gained over the past 10 years to enable all of you to be successful in everything you do. And this feeds into the broader mission of Rosalind. So at the highest level, Rosalind is a unified platform for multi-omics data analysis. But what drives all of us at the company, our mission, is to accelerate the great work that you do to improve human wellness, health outcomes throughout the world. So that's what got me into science originally. And based off of my experience over the past 10 years, there's a lot I can share with all of the nCounter users to help you in your basic science, in your drug development, and just improve the lives of all of us. So that's why I'm here. The objectives for this masterclass are shown in this slide. Primarily, I want to solidify for you, whether you're an individual researcher or represent an institution such as a biotech or a pharmaceutical company, I want to solidify for you the synergy between using both nCounter and Rosalind together. Alone, each one stands alone fantastic, but together you unlock some key value and insights that you don't get when they're not paired together. So as I go through each session in this masterclass series, my main goal to explain to you is what are the basic principles of nCounter data analysis and how do the algorithms work? If you're not mathematically inclined, don't be intimidated. nCounter data analysis is very simple. As long as you can understand arithmetic and basic algebra, you will be fine. And I'll explain everything in plain English. So hopefully it'll be very intuitive for you. At the end of this session, after all 6 sessions actually, I want to make sure I increase your confidence to make sure you're analyzing your data accurately and interpreting your results accurately. And by doing so, you're just going to open up new insights in your analysis that you might have missed otherwise, unless you had this training. So the session we're talking about today is focused on custom pruning of low expressors over the next 2 months or so, we'll be diving into normalization, quality control, pathway analysis, self-profiling, and meta-analysis. But the focus today is on one of the very first steps in all of the analysis, and that's custom pruning and low expressors. It's not flashy. It's based in some basic math and algebra and gene selection, but it's important and it sets the foundation for successful analysis afterwards. So in my experience working with users such as yourself and researchers and scientists, I observed there's 3 main benefits of nCounter technology. I bucketed these into 3 main categories: speed to results, just the elegant simplicity of how things work, and the digital precision. I don't want to belabor these points, but if you've used nCounter, I think you probably appreciate these very much. Speed to results: if you're doing the experiment in a lab, 15 minutes of pipetting, you use automated instrumentation, you can go from sample to data in less than 24 hours. So that's fantastic. The simplicity and elegance of the chemistry really sets the foundation for that digital precision, which is that last bullet point. Long story short, because things are so simple and they've minimized the technical variability, you get very precise, very reproducible data with best-in-class performance with the graded RNA samples. And if you're working in drug development, you're very aware of these types of samples, FFPE samples, and how challenging they can be for gene expression technologies. So that's what I observed over the past 10 years. I think you probably appreciate those if you have used nCounter before. But when it comes to the data, I love the data. It's fantastic. The raw data, the RCC files are small, so you can transfer them on a USB thumb drive. They're very easy to store. You're not looking at gigabytes or terabytes of data. The data is intuitive. It's a single molecule counting platform. So what you see is what you get. It's just single molecule, single counts of every gene. Processing the data is very fast because the algorithms are very simple. You don't need intense computation. And this last point here, easy to understand algorithms conceptually and mathematically. I want to dive into that today, specifically for custom pruning. Now, if you haven't used Rosalind before, this is an overview of the types of modules you might see once you log into the platform. These show some of the highlights for our nCounter gene expression technologies. We'll see some of these as we progress over the next 2 months. We'll see quality control in more depth, pathway analysis in more depth, cell profiling. But I want to highlight for you is that the reason we're doing this masterclass now is because we just released a whole host of new updated features for nCounter analysis. We call this our advanced analysis. So we have new customized pruning, customized normalization. We've enhanced the pathway analysis experience. We've embedded our deep intelligence and knowledge into the nCounter analysis to allow you to get more information out of your targeted panel. And of course, you can use meta-analysis, and we've updated cell profiling. So from my perspective, over the past 10 years of using various types of ways to analyze nCounter data, Rosalind is at the forefront. It is the place to analyze your nCounter data, whether you're an individual researcher or you represent an institution. It fosters collaboration more seamlessly than anything I've ever seen. So let's get to some of the key insights for deep pruning. Before I do that, I just want to say that together, Rosalind and nCounter represent a complete end-to-end solution for rapid discovery and deep insight. Again, you start in the lab with 15 minutes of pipetting, overnight hybridization, get your data in less than 24 hours, and that feeds directly into Rosalind, where you can extend the utility of your targeted panel using Rosalind Intelligence, Rosalind Knowledge, enhanced pathway analysis to get as much data possible out of your targeted discovery. So my last slide, and then we're going to jump into some data, is why do we want to prune low expressors? If this is a new concept to you, I'm going to talk to you about this in depth, why we're doing it. A few bullet points came to mind as I was thinking about this. The first is that you're removing these low expressors to maximize the accuracy and robustness of your normalization. And therefore, by doing that, you can improve the accuracy of your interpretation. And you want to ultimately increase the confidence that your data are accurate. So you want to have confidence to publish them. Or if you're designing follow-up experiments, you want to make sure that your foundation of knowledge is accurate for these follow-up experiments. So that's the slides. Let's jump into the platform and some of the details of pruning. So give me 1 second here as I transition. I'm going to log directly into Rosalind. Okay. So I've logged into the Rosalind platform. If you're a user of Rosalind, you've probably seen this screen before if you've done nCounter analysis. If you're brand new to the platform, this is one of the steps during setup of an experiment. So we have 2 paths that you encounter when you import your RCC data and start analyzing your data in Rosalind. There's this default path and the advanced path. Traditionally, everyone has gone through the default path. This contains a nice set of recommendations on how to prune your data, how to normalize your data, and give you this nice experience through the default path. And that's what we've done historically. But what's new is this advanced path. This is available to all of our paid subscribers, our academic subscribers, our enterprise professional subscribers. And what this enables for you is to customize the pruning of the low expressors, customize normalization as well. And that really sets the stage for making sure you have confidence in the accuracy and interpretation of your data. So from my perspective, having these customizations is, it's a great feature to have, and I would definitely use it in all of my analysis. So the way that this looks is I'm going to click on next, and it's going to take me through a wizard type interface, step by step on how to analyze and set up my nCounter experiment. Now, when we were thinking about developing this from scratch, I kind of looked back on what I've observed over the past 10 years, other types of software packages I've used to analyze nCounter data, and I wanted to challenge the status quo. I didn't want to take anything for granted and pick apart step by step what should be done, how it should be done, and the order it should be done. So when we developed advanced analysis here, this advanced workflow, what we did is we reversed the order of the traditional housekeeping gene normalization and pruning. So if you've done nCounter analysis in the past, you have most likely selected your housekeeping genes first and then pruned the data, removed the low expressors. However, what I observed is that in certain situations, this allows some low expression housekeepers, some housekeepers in the noise, to actually enter the normalization process. And I think everyone's eyes are probably opening right now, because obviously you don't want these low expression probes, you don't want this noise data to be involved in your normalization. First, you want to get rid of those and then perform your normalization. So that is why we switched the order. It was very deliberate. We put pruning before normalization, so you can make sure you have very robust housekeeping genes prior to your normalization step. So it's all hidden in the background. You would never know this has happened, but this is kind of the intelligence of the Rosalind platform. We make these decisions to help you and guide you to get the most accurate data you can. So that's a big step. And then let me dive into it. So I'm going to click next. And again, traditionally, you would have seen housekeeping gene and normalization first, but now we're starting with pruning because it gives you a more accurate data analysis. And that's everything. That's all that we want. We want to increase the accuracy of our data. So let's go through this advanced step. And we come to this interface here. Don't worry about reading these instructions up here. I'm going to explain all of this verbally. I'm going to walk through this entire interface as well. So if you've done this type of thing before and perhaps other software packages, what you would see is basically two sliders here. The only thing you would see in other solutions might be a way to adjust a low count threshold and a way to adjust a sample frequency threshold. What we did at Rosalind is we realized and recognized that by changing these thresholds without having visibility into actually the effect on the genes themselves, then you're basically flying blind. You might set up your experiment, you might pick a count threshold of 16 and a frequency threshold of 85, run your analysis. You might get to the end of your analysis and realize, oh, I might need to go back and set up this entire experiment from scratch because I might have picked the wrong values. Here in the Rosalind platform, we allow you to do all of this in real time, so you get the right setup, the right analysis from the beginning. I don't have to have this iteration of going back and forth and playing with these numbers. So that's the beauty. You can change these pruning thresholds and see the exact effect on your genes, and pick and choose exactly which genes you want to include to make sure you're keeping your key targets in your analysis. So all that being said, let me make this a little more simple and break down what exactly custom pruning is and custom removal of low expressors is. So I'm going to kind of go underneath the hood of what this interface does and explain it to you in a very kind of simple step-by-step fashion. Because what I found is that the more you understand these concepts, the better outcomes you have in your analysis. Yeah, I mean, it kind of makes sense. If you know what you're doing, if you understand why you're doing things and how you're doing things, then you just get better outcomes. So that's the purpose of this session right now. So let me go to Excel, and I just want to show you some basic calculations. I love Excel, but it's not great for data analysis, as you know, but I want to show you how the calculations are done. So to do that, what I did was basically click on this Export Probes button, and I downloaded the raw data, and I can do a deep dive on all my controls, all my endogenous genes, and all of my housekeepers. So that's going to look like this. I downloaded the raw data directly from the Rosalind platform. This is kind of a hypothetical experiment. I have 10 samples in this hypothetical experiment, labeled samples 1 through 10. If you know the nCounter data, you know each sample contains 6 positive controls, which I'm not going to talk about today. I will in future sessions. But we have 8 negative controls within each sample. And then we have a series of endogenous genes and housekeeping genes in all of the samples. What I really love about the nCounter data is that we can empirically determine the limit of detection. Many technologies don't have these built-in negative controls, but by including the negative controls in each individual sample, you can empirically set what is the limit of detection. And that allows you to figure out which data points you want to include in the analysis and which genes you want to exclude. So I just love the negative controls. It's a great thing about the nCounter technology. So to start this kind of exploration and explain conceptually what I mean by pruning of low expressors, I'm going to move to the next tab. So I'm just going to take this data, we're going to move over to the next tab, and I did a few calculations that I want to show you. And that's shown here. So let me break this down for you. I'm going to calculate what's called the limit of detection. Limit of detection is established in order to figure out what data points are present in the analysis, what are true signal, and we want to separate those from data points that are below the limit of detection, which are below background, which are in the noise, which are not real signal that we want to ignore and remove them for our analysis. So again, the purpose of establishing this limit of detection is to separate the true signal from the noise. That's what it is at its fundamental level. Now, when I use the term limit of detection, that means something very specific in NanoString terminology. Limit of detection is one of the technical specifications of their assay, and they define it as the mean of the negative controls plus 2 standard deviations. So I'm going to break down exactly what this means for you. So to establish the limit of detection, let me look at sample number 1 as an example here. I'm going to take the 8 negative controls in this sample, and I'm going to calculate the arithmetic mean. For this sample, it's 7.63. I did that exact same calculation for every sample here, all 10 samples. If you love Excel and like getting into the formulas, I'll pause here just for a second. This is the way to calculate the average of all the negative controls for sample number 1 here. Next thing I did, I calculated 2 standard deviations of the negative controls in sample number 1. So that's represented here. If you want to see the formula, hopefully it's big enough for you to see. It's just 2 times the standard deviation of all 8 negative controls for sample 1. And then, of course, I copied this formula across all 10 samples. Finally, to calculate our limit of detection, I just added these 2 values together. It is simply going to be the mean plus 2 times the standard deviations. And for sample number 1, that is 14.71. And again, I calculated this for all individual samples. Now, of course, you can do this formula all in one cell and make it much simpler, but I broke it down step by step just to hopefully explain it to you a little more clearly. So what I just did, I calculated a specific independent limit of detection for each individual sample. You see there's some variation here. So this is 14.71, 19.32, 15.77. This one's down at 8. Some variation is normal. This just reflects the natural technical variation from lane to lane in the cartridge. And I just want to encourage you, when you look at these differences, don't overinterpret them. Like you might see this as this is 8 and this is 18.60. This is not actually a big difference, even though it might look like it visually, 18 versus 8. But if you think about the dynamic range of the assay, it's about 5 to 6 logs of linear dynamic range. So some of your low expressed genes might be, I don't know, 40, 50 counts, and some of the high expressors might be 100,000. So it's a really broad dynamic range. So if you're thinking about the difference between maybe 18 and 8 or 12 and 18, you're really splitting hairs, and it's not really going to have an impact in the end. So if you're not familiar with the natural variation of the limit of detection, this is normal and it's expected. Okay, so the next thing I did, I wanted to summarize across all of our samples, what does this limit of detection look like? So its minimum is 8.7, its maximum is 19.32, and I simply took the average across all samples, and that's what I called this experiment-wide limit of detection, something called 14.30. Remember these values, because we'll see these again when we get back into the Rosalind platform. We calculate these for you and display them to you so you can see them without having to go through all of this Excel math. Okay, so hopefully that explains limit of detection, how it's calculated. So the next step is to figure out what data points you want to keep, what data points we want to exclude. So as an example, we have this first row here of genes, C4BPA. What we want to do conceptually is just compare this data point, 30, versus its LOD, its limit of detection, for the same sample, which is 14.7. So 30 is greater than 14.7, so this is a positive signal. We want to include this in our analysis. As another example, let's look at this data point here. So C4BPA in sample number 6 has a raw count of 1, which is clearly less than the limit of detection here of 14.55, so this data point is in the noise. So this is the basic conceptually what you're doing. You're comparing all of these data points one by one to its respective limit of detection in the same sample. So I'll have some figures that show this, and if you're not having trouble visualizing this, well, that's conceptually what we're doing, comparing the raw counts for each gene versus the sample-specific limit of detections. So before I go and show you those figures, I want to explain, why is the limit of detection the mean plus 2 standard deviations? Does that make sense to you? Do you understand why this is happening? I want to give you some basic grounding, just at a very high level, why this is actually the limit of detection, and I think that's going to give you some more confidence in what we're looking at. So I'm not going to teach you statistics. Certainly not going to do that at all. I can see the eyes glazing over right now. No one likes to learn stats, but it is important. So I want to introduce a basic concept here. This is the normal distribution. You've probably seen these types of bell curves before. Don't want to get too much into the weeds, but what we're seeing here is something called a standard normal distribution. What this represents is a population mean of 0. That represents mu here. This is the population mean. In the standard normal distribution, the mean is 0, and the standard deviation is a unit of 1. So that's what we're seeing here. Mu represents mean of 0, and the sigma is standard deviation. So this is 1 standard deviation, 2 standard deviations, and 3 standard deviations. So what I want you to walk away with from this long explanation is that there's a nice rule of thumb here. Sometimes it's called the empirical rule, but it basically predicts in this type of normal distribution, where do your observation falls in relationship between the mean and the standard deviations. So what I mean by that is that if we look at the green region, so this represents all the data within 1 standard deviation for the mean. The observations are the data, or this should represent 68% of the data, essentially. If we think about 2 standard deviations, so that's all of this surface area, including the yellow from here, all of this green to the yellow here, so all of this region, that should account for 95% of the data or the observations. Now, when we go up to 3 standard deviation, that gets even more inclusive. It represents approximately 99% of the data from 3 standard deviations, all the way from down here, all the way up to here. So when we think about this in terms of the NanoString data, we can kind of compare what is the distribution of the NanoString negative controls versus the standard deviation. And I think that's going to be illuminating on why we are using this calculation of mean plus 2 standard deviations. So that's shown here in this next slide, or this next image. On the right here, this is a data set generated by NanoString. So I dug into my treasure trove of all these old public experiments and public technical notes that I had just sitting in my archive. And I remembered I had this one figure, which would be perfect for this type of seminar here. And this shows you the distribution of the negative controls for a NanoString experiment. And hopefully you're seeing a lot of similarity to that normal distribution on the left-hand side. There's a couple caveats I want to show you here. The first is that this is a data set generated from 288 samples across 10 different code sets and with different numbers of genes. So just a handful of genes all the way up to perhaps like 770 or so. So it's a kind of a variety of different data sets. And what we're seeing is that it's approximately normally distributed. I'm not going to do a formal statistical test to look at the fit of this distribution and see if it's normally distributed. That doesn't really matter too much, but I think just approximately it's normally distributed. And the last caveat here, which I want to explain, is that each data point here, this actually represents 1 sample. So this circle is the average of the 8 negative controls for 1 sample. So this curve we're looking at is not individual negative controls, it's just the average of negative controls for all of these 288 samples. So that's a caveat I wanted to explain, so this figure is not misinterpreted. In the end, it probably doesn't matter. I think what I wanted to show you is that the negative controls for NanoString nCounter gene expression is approximately normal. which is interesting if you didn't know this was true. And that adds value to this limit of detection calculation of the mean plus 2 standard deviations. So essentially what this sets up for us is that there's basically a 95% confidence that any data point above the limit of detection is a true positive or a true signal. Now, this isn't like a formal confidence interval calculation. This is just kind of like back of the envelope math, conceptually how I think about this. But yeah, any gene, any data point above the limit of detection, there's a 95% confidence that that's a true signal. So that's really interesting to know. I think it gives you confidence that the limit of detection establishment is a really important thing to do, and it just gives you confidence in your data. Okay, so that's this long-winded explanation about the distribution of the negative controls. It's something very few people know about in my experience, so I wanted to share this information with the broader community because I think it is really important to know. Okay, so that being said, we can go back to our limit of detection calculation. So as I said before, we want to figure out kind of on a gene by gene or data point by data point basis, which genes are above the limit of detection, which genes are below the limit of detection. So to do that, I illustrated that in my next tab, which I'm calling Pruning here. And this is the same data, so there's nothing new here. I have the same calculations I did, this sample-specific limit of detection for each individual sample. And then what I wanted to do to show you is, in order to kind of create a frame of reference for you, is that there's 3 main classifications of probes that you need to think about when you're thinking about pruning of low expressors. The first set is right here. I'll just show you all 3. This is the first set. Basically, the gene, the probe, is above background, above the limit of detection in every single sample. And I did include here separate lines for endogenous probes versus housekeepers, because as I said before, it's really important to make sure you don't have below background housekeepers in your normalization. So this is something I'm going to mention again when we get back to the Rosalind app. But it is important to note that you can make a distinguishing here between endogenous versus housekeeping genes. So clearly, CD24 as an example, this data is clearly above the limit of detection relative to all of these values here. That's kind of a no-brainer. It's obvious. Same with this gene here. It's not as high as the other one, but it's still above the limit of detection. And the same for these 2 housekeeping gene examples. Clearly, this data is above the limit of detection. So we want to include all of these genes, all these probes in our analysis. The next example here is something kind of in the middle between this and no expression, between samples have universally expressed and samples where there's no expression. There's this middle ground. What I've done is I've colored data points that are below the limit of detection in this pink color. So when we look at C4 BPA, these white data points are going to be above the limit of detection. So again, that's in reference to these data points here. These in pink are actually below. So these values here, these 3 values here are less than these, so that's why they're pink. And then these 2 genes are just an example of you get this mixture. This is common, not only in NanoString, but in all gene expression technologies. So we need a robust strategy to figure out how to handle these situations. And again, I highlighted housekeeping genes here because does it make sense to use this housekeeping gene in normalization if this data point is below background? That's a question I'm posing to you. I would say probably not. Why would you want to normalize with this data point, which is noise? We probably want to remove this one. If you're doing housekeeping gene normalization first, there's a chance that this gene might enter the normalization process, which is what we want to avoid. And again, this is highlighting why I wanted to put pruning before normalization because of situations like this. Finally, the last class here, the last situation, is a no-brainer as well. We look at IL-1A, this gene here. Clearly, it's below the limit of detection in every single sample. Let's get rid of it from the analysis. So let me show you these visually, because if you're not a spreadsheet person, if you're not a number person, I want to add visuals if that helps you understand what I'm talking about. So here, this graph represents, for samples 1 through 10 on the x-axis, what is the limit of detection. So for example, sample number 1, the blue bar is the average of the negative controls, and then the whisker, the error bar on top, this represents adding 2 standard deviations. So this value, pretty close to 15, this is the limit of detection for sample 1. So again, you can see some variation across all 10 samples. And again, I just want to make sure that we're not splitting hair here. That's a tongue twister. But I want to make sure there's really no alarm bells going off, because this one is close to 19 and this one is close to 8 or 7. In the grand scheme of the whole dynamic range of the assay, from let's say 50 counts to 100,000 or more counts, a difference of 8 or so counts here in the limit of detection, probably not going to have an impact. So don't overinterpret these differences. Okay, so that's how the distribution of limited detections look. Below that, I want to show you the different use cases or the different classifications of genes we want to think about for pruning. So this is the clear case, the no-brainer, where we have this gene TNFR-SF14. I plotted its raw counts here on the green line versus the limit of detections. And clearly, this gene is well above background across all samples. Obviously, we want to include this in our experiment. We want to analyze it. That just makes sense. The other clear no-brainer use case is this one here, where we have interleukin-5, the raw counts for interleukin-5 plotted versus the limit of detection for each sample. Clearly, this gene, this probe, is below the limit of detection across all 10 samples. We want to get it out of our analysis. There's the risk, if we include this in our analysis, then you might actually conclude that it's differentially expressed across your different treatments or sample groups or disease states. So obviously, we want to get this out of the analysis so it's not misleading. We don't get misled in terms of single gene differential expression or in pathway analysis. We want to get rid of all these genes prior to pathway analysis as well. Oh, and cell profiling. So yeah, it has a lot of implications. If you don't get these genes out of your data set, it impacts differential expression, pathway analysis, and cell profiling. And the final use case, what I want to show you is this down here. So I'm going to pause just for a few seconds and challenge you to think about, do you want to include this in your analysis? The gene is C4BPA. It's above the limit of detection in some samples, as indicated by the orange line, but below the limit of detection in other samples. So how would you handle this traditionally? What would you do to make sure you're getting these genes out of your analysis? Or do you want to include this gene in your analysis? What strategy would you use to handle these genes? It's a challenge. This is happening all in the background that Rosalind is going to guide you through. But this is the challenge that we're all facing, whether or not we realize it or not. It's really important to make sure we have a robust strategy to handle these genes. So I'll pause for a few more seconds. I'm not going to call on anyone to volunteer their opinion on how to handle these. I'm not going to have a pop quiz, but just think about this. Like, what would you do? What are you currently doing to handle these genes? Okay, so the solution, the best solution, is what we do within the Rosalind. We have our advanced pruning platform, and we offer you a very robust, mathematically principled way to handle these types of use cases in a systematic fashion. And the beauty of it is, after you make your systematic selection across all 800 genes, then you can go one by one looking at your key genes of interest to make sure those are actually included in your analysis. No other software had that capability. So it's a really nice advantage of doing this type of thing in Rosalind. So I'm going to go to one more math slide and then back to the platform, and I'll pause for some questions. So we should be wrapping up here in just a few minutes. Let's talk about what happens. Well, let me show you quickly back to Chrome. Oh, I got back just in time before I got logged out. So what's happening in the background in this interface? I want to show you that in Excel, not to overwhelm you, but just to kind of illuminate what's happening. And that's shown here. So don't get overwhelmed with all the data, all the calculations. I'm going to explain it pretty simply here. The data at the top is the same. We have the same negative controls for our 10 samples. We have our same limit of detection calculations for each individual sample, and our same descriptive statistics. We're going to see these in just a minute or two within the Rosalind platform. So we calculate these for you for every individual experiment that you run. So this is a pretty cool feature. And then what I've done below is I created this little, I don't know if I want to call it an app, but this little kind of tool within Excel. It took me quite a long time to build this, but this basically is showing what's happening in the Rosalind backend. This is nothing you would ever want to do for each individual experiment you run. because you'll just be burning hours and hours and hours, and there's typos, and like it's just a nightmare. But I want to show you what's happening so you can understand conceptually what Rosalind is doing for you in a very simple fashion. So what I did, I created a tool where down here for each individual gene, let's look at C4BPA, we are color coding in pink all of the data points that are going to be below our limit of detection. All of the data points in white are above. I created these two boxes here, which I can complete and customize myself. This is going to be mimicking the type of functionality we have in the Rosalind platform. We can establish our low count threshold. Now, instead of using the word limit of detection, I decided to use the word low count threshold because it's a little more generic. Again, as I said in the beginning here, the limit of detection has a very specific technical meaning in terms of the specifications of the assay. So in the Rosalind platform, you'll see the words low count threshold because that gives you the freedom to kind of adjust this value. And it's just a little more of a generic term, so it's more appropriate in that sense. So what I can do in this dashboard here is I can change this threshold. So instead of 15, let me change it to 30. And what you can see is that down below, we are color coding different genes in pink. So as you can see, as I increase the value, let's, I don't know, 50, something really high, a lot more data points. Let me go to even like 100. A lot more data points are going to be below background. So this is one way to customize which genes you're removing. Let me set this to, let's use a value of 15. That's approximately this average LOD across all samples. And then what I'm showing you here in Excel is this column, is that when we look at this first gene, C4BPA, how many of the samples are below this threshold of 15? So that's all this calculation is. If you want to see what it is in Excel, it's shown here. I'll keep it there for a few minutes if you're curious. But basically what I'm doing is just counting the number of pink data points. So for C4BPA, our first gene here, 5 of the 10 data points, 5 of the 10 samples are less than our threshold of 15. Okay, so hopefully that's clear. I'll explain that one more time. I'm just counting the number of these samples, the number of these data points in pink that are below our threshold of 15. And in this example, it's 5 of the 10. And expressed as a percent, that's 50%. So in sample number, or in gene number 1, 50% of our samples are below our threshold of 15. And we can do this calculation for every single gene. So C7, the next gene, 0 samples are below our threshold of 15. So our percentage here is 0. Here's one that's 100. Here's one of the other use cases. So clearly we want to get rid of this gene from our data set. Now the other component to this is frequency threshold. This is where it gets nice and customizable. This is where the power really comes in. And I'm going to move a couple of my windows here. 1 second. Okay, so this frequency threshold comes into play is we want to ask the question in what percent of samples, or what's our threshold in terms of sample frequency where we want to actually remove the gene from the data set. This is the question. Let me just refresh your visual memory here. This is the question I was asking here. How do we know whether or not we want to remove this sample? The strategy here is to base it on sample frequency. How many samples or what percent of samples is this gene below the limit of detection or below our threshold? That's how we control that. So we can do this, of course, in the Rosalind platform, but just to see what it looks like in Excel, if I make this, let's say 10%, then if I scroll to the right, what I'm showing you is, this is a very stringent threshold, just so you know. The lower the number this is, the more stringent it is. We're actually removing 231 genes from the data set. If we make this more permissive, let's say 85%, which is the default in Rosalind, then it's 82. So this is how it works in the background. This is how the math is done. Hopefully this helps you understand what we're doing in the Rosalind platform when we go back to it here in just a couple of minutes. Just to reiterate before I go back, we can establish our count threshold. Typically, this can be based off of the limit of detection for the negative controls, or I can customize this to any value I want. If I'm an advanced user, I can set this to any value I want. And I can also customize the threshold frequency. So these two values, the count threshold and then the frequency threshold, work together to figure out which gene should be removed. So that's kind of the gist of it. I know it kind of gets a little complicated, but as one more example, this gene here, what is this gene? IL-13RA2. It's below the negative controls, or below the threshold at nine samples or 90% of the samples. Therefore, I flag this as yes. Should it be pruned? Yes. So we can do that in Excel for each sample one by one, and then eventually create a list of genes we want to remove. So it's a very laborious, very error-prone process that I wouldn't recommend to anyone, but this is what's happening in the background within the Rosalind platform. So let me switch back there and bring us home, show us how to work with this in Rosalind. Okay, so we're back to Rosalind. Now, everything we just learned is going to help us in this page here. So you'll see we have this low count threshold that we can change. We just did that in Excel. We have the sample frequency threshold that we can change that we just did in Excel. And based off of these two thresholds working in combination, Rosalind tells us that there's 60 probes that's recommended to remove based off of these thresholds. When we look at the table on the right, this middle table, this contains all probes in the panel in your code set. So I can sort this table. So let me sort by probe name. So I can sort alphabetically. I can sort by category. So we can have our positive controls, our negative controls here. We have our positive controls here, our housekeeping genes, our endogenous genes, average counts for each probe across all samples, and our percent of samples less than our count threshold. And then on the right-hand side, this is where it gets really nice and powerful, is that this is the list, this is the view of all of the genes we're removing from the data set. So I want to talk about strategies about how to use this page, but that's the general layout. So if you don't, if this is intimidating to you, if you're not comfortable with changing the thresholds, you don't have to. I would say that in most cases, the defaults are going to be really good to use. So if you're not comfortable, you don't have to change the threshold. You don't have to change the frequency, use the defaults. But I would encourage you to further explore the data in the middle if you don't want to change the defaults. So the way that I would do this, if I came here the first time, I would not change these thresholds, maybe because I'm not an advanced user. I don't understand necessarily how I'm impacting the data. So let me just use the defaults because that's going to be really robust in most cases. But I'm going to come into this middle table to look at all of my probes, all of my genes in this panel. And I'm going to sort by number of samples less than threshold. And so what Rosalind is telling you is that all of these genes color-coded in this pinkish, purplish, pinkish peach color, which is in this case 60, these are the ones recommended to remove. These are essentially noise. We want to get them out of the analysis so they're not adversely impacting normalization, differential expression, or pathway analysis. So what I would do, I would sort by this value here, get all of these 100% samples at the top. Clearly this gene USP9Y has an average count of 4 across all of my samples. It's less than our threshold in 100% of samples. This is pure noise. We don't want this in the analysis. So if this gene has this red arrow, that means it's going to be pruned, it's going to be removed, and it's going to be in this list on the right-hand side, all the way at the bottom down here. But what's really interesting to do is to scroll down here, and I would recommend coming and looking at this border region here. So this is the border region where everything above this is going to be removed, everything below in white is going to be included in our data set. So I would recommend just coming through here and looking at these genes along the border. Do you recognize any of these genes? Are they going to be potentially important in your analysis? Are they part of your pathway of interest? Are they part of your signaling pathway? And what's really cool here is that, let's say, I actually want to change these frequency numbers. Instead of 85, let me put this up at 90. And what you'll see is that now we've just updated. So now the barrier, the threshold that I just mentioned, is going to be up here. It's at 90, so it's between 93 and 86. So you can visually see how you're impacting the recommended removals when you change these numbers. So even though you might not have done this before, I would encourage you to play with this. It's really powerful. I think you use it just for a few minutes and you understand what you're doing. I would not be intimidated. You can always just change this back to 85%, which is the default. There's no risk there at all. So definitely play around with it. The other strategy I would recommend, when you look across like these border regions and which genes are here, would just be to sort alphabetically. And so let's say I'm a scientist. I've studied cholesterol biosynthesis in graduate school and in my postdoc. So I'm really interested in these enzymes that form cholesterol, tyrosine hydroxylase, PNMT, DDC, all these different enzymes. So I know them in my mind. I'm a specialized biologist. And here, Rosalind is really empowering the biologists to leverage their domain expertise to come through this list, sort it alphabetically, and make sure all your key targets of interest are included. So I'm scrolling here, and let's say I'm really interested in maybe the complement pathway, and I see, oh wow, CA8 is set to be pruned. This is actually an important gene for me biologically because I'm studying this area. So let me just do this. Let me just make sure it's included. So this is the power and the beauty of the Rosalind interface and having this very granular gene level view. You want to make sure you include all of your key targets of interest. So that might be a good place to stop for some questions. Let me see if there's anything more that comes to mind here. Yeah, I might hand it over to our wonderful moderators to see if there's been any questions that have come in that I can address.
While Cookie's looking through the list, I'll just remind everyone at this point, if you have a question, go ahead and click open the question tab there in your GoToMeeting interface, and you can write up any questions that you had in mind. Thank you for those that held those, and you can enter those now, and we'll do our best to address those questions. And on that note, Cookie, did I give you enough time to pull together the questions that have come in today?
Yes, thanks, Tim. There are a couple. The first one is, what are your recommended cutoffs to use for the pruning?
Yeah, that's the most common question that we get it all the time. And there's good reason, because if you haven't done this type of thing, you don't really have a foundation of, why are you choosing this? Why are you doing that? And again, I'll refer back to kind of the brilliance and the elegance of the NanoString nCounter data and the fact that there's 8 negative controls in each sample. And that allows you to empirically set that limit of detection threshold. So in terms of your count threshold here, if I was going to come to this page the very first time, I didn't know what my count threshold would be, I would rely on the Rosalind defaults, because the Rosalind defaults is going to be that limit of detection calculation, which is empirically determined. And there's a lot of great data backing up that approach. So that's what I would do for the threshold. In terms of sample frequency, this gets a little more nuanced because it's going to depend on your experiment. By default, the value is going to be 85%. This is going to be permissive, so it's going to let more genes into the analysis rather than being too restrictive and removing more genes than you want to have removed. So we decided to be more on the permissive side with the default. But I would say, think about your experimental design. How many different, perhaps, sample groups do you have? Let's say you have one group of samples control, the other group of samples treatment. So you have 2 treatment groups. In that scenario, perhaps a threshold of 50% would make sense. That allows for the fact that any given gene or any given probe could be totally not expressed. It can be completely below backgrounded in the noise in one group of samples and expressed in the other. So with 2 groups of samples, maybe a 50% threshold makes sense. Let's think about another scenario. Let's say we have 3 groups of samples. You might have a control group, you might have treatment 1 and treatment 2. So in that scenario, you have 3 different groups of samples, and you might have a threshold of that case 2/3 or 66%. And that's going to allow for the fact that the gene might not be expressed in 2 of the sample groups, but would be expressed in the other, or the last third. And I'm just going to hammer this point home. One more example. Let's say you have 4 different sample groups: control, treatment 1, treatment 2, treatment 3. In that case, maybe 75% is the right cutoff, and that's going to allow for the fact that the gene might be expressed in only one of the 4 groups. So hopefully that gives some idea of how to think about the sample frequency. It's virtually impossible to kind of recommend this ahead of time without knowing about the experimental design, because everyone has different experimental designs, different number of groups, different number of biological replicates. So that's what I would say there. Hopefully that answers the question for the group.
Thank you, Ryan. We have one more question. It says, can we do percentile ranking of the gene?
Percentile ranking. Well, in the Rosalind platform, we can certainly scroll to the top here. We can sort by average counts. So this is your gene COL3A1. This is its average raw counts across all samples in this experiment. So you can rank by average counts. It's not going to be like a percentile based type thing. If you needed that type of approach, you could certainly do that type of thing in Excel, and that's why let me go back. That's why we allowed this download button. So you could download the data if you wanted to do some additional type of ranking or selections, some kind of more advanced, sophisticated measures. You could do that in Excel, and then you could certainly come back into here and just make sure all of your genes are included or excluded according to your preference.
Awesome. Thank you.
We have a couple more questions that have come in. So Cookie, if you want to grab those.
Sure, yeah. One more question is, does the low count threshold use the same number for all lanes in the cartridge, or does it change lane by lane basis? Example affected by LOD.
That is a good question. Yeah. So the very first time you come to this page, let's say you're setting up your new experiment, this value is going to be set. And what it's going to represent a specific limit of detection for each individual sample. So your limit of detection, your low count threshold, is going to be unique and independent for each sample. That's what happens the very first time you come to this page, and that's the default. Those are those sample-specific calculations I showed in Excel. And then if you hover over this with the tooltip, can't remember if I showed this, but that's what's stated here in the first sentence here. So by default, when you come to this page the first time, calculate a unique threshold for each sample independently. And it's that same calculation I showed. It's the arithmetic mean of the negatives plus 2 standard deviations. Then, excuse me, then we give you those summary statistics, which I showed you in the Excel spreadsheet. We tell you here what is the minimum of the LODs across all your samples, what is the maximum, and then what's the average. So all those calculations I showed you in Excel, those are shown here. And the default, the very first time you come here, it's the unique LOD for each sample independently. Now, once you want to start changing this, then at that point, it applies a universal single threshold across all samples universally. So if I come here the first time, I have my unique LOD for each individual sample, and I change this to, let's say, 20, then at that point, my threshold is exactly 20 across all samples. So hopefully that adds the clarity, but that's a really good question. I'm glad that one was asked.
And Cookie, I'll help with the next one. This is a great question too. So this person says, thank you for the super clear explanation. In my case, I already have an Excel sheet with the normalized counts. Is there a way to upload that sheet to start visualizing the data? In other words, to get the heat map, the volcano plot, etc. Or do I need to start with an RCC file?
You can do either one, according to your preference. I mean, I would certainly recommend starting with the RCC files because that gives you the advanced functionality with custom pruning, custom normalization. However, there's a lot of previously analyzed data sets just like this one. And in that case, we enable the researcher to just upload the normalized data and then take advantage of all the downstream intelligence, the enhanced pathway analysis, etc. So both approaches are valid. I don't know how big this data set is. So if it took a lot of work to get normalized, you might just want to stick with that. But my recommendation would just be start with RCCs if it's not too much additional work and just take advantage of all the new features.
Fantastic. And Cookie, did you get the one on low count threshold already?
Yes, yeah.
Perfect. And it looks like there is another one that came in, and I'm going to ask one more that I also saw while you're looking at that. Looks like the last one that just came in. So Ryan, there's one here that says, on my system, I'm only seeing that I have the default and I'm not able to click on advanced. So could you address that question?
Yeah, so the advanced workflow is available for our paid subscribers. So these are academic students, researchers, academic teams, professional nCounter users, enterprise users. So if you're currently on a sponsored subscription from NanoString, you do not have access to that basic workflow, or I'm sorry, you don't have access to the advanced workflow. So that's probably what's happening there. We can certainly talk more offline if you want to, learn how to get access to the advanced workflow, but that would be the most likely cause there.
And actually, to that end, Kuki, why don't you quickly post on the chat, or in fact, while you're helping with the next question, I'll post on the chat a link folks can click that will give them a chance to set up time with Ryan's team so that you all can ask more questions and also potentially find out about how to get access to the advanced functions if you don't have access right now. So while you're reading the last question, Kuki, I'll go ahead and post that link into the chat field if I can find that.
Yeah, and there was a question around if there will be a recording made available after the webinar. You guys will get an email, so that will definitely be there. So no worries about that one in case you missed something.
And Kuki, I'll add to that. We also have Rosalind Academy, if you are not aware. It's Rosalind. I can actually probably show you since I'm sharing my screen. It's a one-stop resource for everything Rosalind. We're putting a very strong focus on nCounter. So we have trainings there. So you can train yourself by watching pre-recorded full-length videos. We have self-guided trainings if you want to use some demo data set and just work at your own pace. We have live nCounter trainings every other week. We have a knowledge hub, method section as well for all of this stuff. So let me kind of just go there and show what this looks like. And then all of us can have the URL. So I would encourage everyone to come here. If this is everything Rosalind, we're putting a very strong focus on our resources for nCounter. So I'm not going to do a full demo here. I want to make sure we answer questions, but just certainly explore this. All sorts of training options, live, recorded, self-guided, video library, we have support, knowledge hubs. So this is your first resource that I would recommend as well. And the series will be posted here, of course, in our video library.
And I just posted the link for folks that want to request time with the customer experience team, and they can help you with both your scientific questions, of course, and also if you get connected to learn more about how you can access these advanced capabilities and also look at some of the options for subscription upgrades where that's appropriate. On that note, Ryan, I think it's probably time we wrap this up. So thank you so much for taking us through this. Ryan, I'll hand it over to you to do the final close.
Yeah, I want to thank everyone for joining. It looks like we had a really good crowd, which I was very happy to see. I'm looking forward to continuing this series. I really want to share all of this information with all of you because the more you understand the individual steps, the better outcomes you have. So I really hope to see you again in the future in additional sessions. And I appreciate all your questions. I think it was a great benefit to everyone here that joined. So I will see you in the next session. Let me go to
which is
schedule.
Exactly. Thank you, Ryan. I was just about to remind you of the schedule.
Yep, this one here. So this is the schedule. Our next session is May 2nd, and we're going into customized normalization. So I'll explain the details of how normalization works, different approaches, different strategies, and it'll be very similar to the session we just did. So I will see you then. Thank you, everyone.